Now we are on the hour, and I see a lot of people dialed in, so let's get it started. Welcome everyone to Dashu Virtual Journal Club. I'm the president of Dashu, and I'll give you a quick introduction what Dashu is all about. So we are a 501c3 nonprofit organization started about 3 years ago, and we have quite a few strategic partners across the Bay Area, including the American Statistical Association Bay Area Entrepreneur Statisticians, etc. And everything is free in Dashu. I think the only exception is we do annual conferences, international conferences that will require registration fee and sponsorship, and that's where the revenue comes from. Other things we provide to this community is all free, and we have now more than 1,000 members across the world. Everyone here volunteers, so the administrative cost is extremely low. It's just the application fee we file for IRS to keep our nonprofit organization status. So even our CPA, everybody works for free, so every single cent from donation, sponsorship can be returned to you, serve you and the community. And what is this? Why Dashu? So Dashu actually is a pun in Chinese. It means big data, and it also means community. So in Chinese, there's a phrase we say, big trees. You plant a tree, and it will grow big, and the shade will benefit many generations to come. So we're trying to grow a virtual tree and benefit the community, and basically promote research and education in data science. And we organize international conferences, monthly journal clubs like this one, technical seminars, and also soft skill career development events. We are actively seeking members, volunteers, new ideas, and money if you want to be a sponsor as well. So everything you can learn us more from dashu.org. So it should be easy to type into the webpage and you can learn more about us. Next slide, please. So our biggest, one big exciting event coming up is in early November. It's a local event at Foster City. It's Bay Area Biotech Pharma Statistics Workshop. So it's going to, because there are so many biotech life sector related industrial partners in Bay Area, we thought we should have such a conference focused on that. So this is the inauguration conference we're hosting. And actually, if you go to our website, you scroll down, you will see exactly this picture, and you click on it, you will learn more how to register and so on. Next slide, please. Here are just some highlights of this upcoming conference in just about 50 days. So we have 3 keynotes that you can see very prominent. We are very honored to get our current ASA president to give us a keynote speech, as well as co-founder and CSO of Genomic Health, who is a pioneer using genomic information to do personalized diagnostics, as well as the Senior Vice President, Dr. Hu from Nectar Therapeutics, to talking about personalized treatment. And the featured sessions you can see are mostly cutting edge advancement in, clinical trial design and the real applications on real patients that provides clinical impact. So if you want to learn more, just go to our organization, find the BBSW logo, click on it. And if you have questions, I'm more than happy to answer either through the chat box or just, after the talk. Next slide. And we have a virtual journal club. We're trying to do it monthly. And again, if you go to our website, you will see that a blue virtual journal club logo, and click on it. Actually, as long as the speakers agree and we work with speaker closely, we share of our previous journal club content, slides, recording, all free for you to enjoy and benefit. But we understand sometimes the speaker, if it's working for a startup or the institution, have legal concern, so sometimes cannot be shared. But if possible, we share everything. So not only you can enjoy online at the real time, you can also go back and look at some of our old journal club. I think we have more than 10 archived there. And that is the website to that journal club. And as I said, if you go to our main website, find the virtual journal club logo, you can try that as well. So next slide. So today, I'm so excited. We have Dr. Fernanda Pulubriganoff to give us this exciting talk on disease heritability inferred from familial relationship reported in medical record. I want to tell you how exciting that is. Not only I feel it's excited, this is published in Cell, one of the most prestigious journal earlier this year in May, and Dr. Fernanda is the first author, and on the same day, I think Cell Press give a special news release on this topic, and in New York Times, there's an article dedicated to this fascinating research called "Will You Be My Emergency Contact?" Takes on a whole new meaning. So not only is in the statistics and the bioinformatics community, this is an exciting topic. I think it has a huge general relevance to the general population. And Dr. Pulubriganoff is a brilliant researcher. She got an MD and a PhD. So she is a physician, she's a treating physician and an informatician. I'm thinking about that. Doing one is already challenging enough, and she manages to do both. And she published this Cell publication while she was still a PhD student. So that tells you just how wonderful and brilliant she is. And not only she is smart and extremely capable technically, she also has a big heart. So she supported our organization by offering to provide this free seminar to all of us to learn more and interacting with her. So without further ado, let's hear Dr. Pulubriganoff's wonderful talk.
Thank you so much for the nice introduction, Jing. I really appreciate it. Thank you so much for joining us today. As Jing mentioned, we'll be talking about our project in which we estimate disease heritability using familial relationships that were inferred from electronic health record data. So, and during this talk, we'll be divided in two parts. First, I'll describe how we identify the familial relationships using existing data from the electronic health record. And then I'll explain how we used these relationships to study heritability of multiple clinical traits. So electronic health records are used every day as the system of record to support clinical care. In these systems, we collect patient demographics information, such as race, ethnicity, address. We collect medications that patients are taking, reported allergies. We also look at, we have data for immunization history, results of laboratory tests. We have radiology reports, vital signs, billing information, and this list goes on. With federal initiatives such as the CMS Meaningful Use Program, electronic health records have been widely adopted in the United States, capturing information of thousands of patients on a daily basis. However, the records of family members are not currently linked together. So we asked the question, what data could tell us about shared genetic background? We have race and ethnicity data. However, 40% of our records with this information are uninformative, such as other or unknown. But there is a more reliable source of relatedness that is collected in hospital, and this is the emergency contact information. It is part of our hospital processes to collect an emergency contact at the time of admission. Patients are asked to provide someone's name, phone number, address, as well as the relationship between the patient and the provided emergency contact. At Columbia University, we have about 2.5 million emergency contacts, and the majority of the provided contacts are part of the nuclear family, such as mother, father, spouse, child, and sibling. Using this method, we were able to publish this paper at Cell. So if you're interested in looking at details of the method, please feel free to access our article. So when we use emergency contact information during this method that I'll start to describe now, we match the first name, last name, phone number, and zip code to the list of patients that we have at the hospital. We only consider a match if we identify a unique patient that matches using any of the elements or any combination of these elements. The assumption here is that families oftentimes use the same healthcare system, and therefore the emergency contacts provided could also be patients in our patient list. Once we applied this method, we obtained 1.1 million matches. Some of these matches have informative relationships, such as mother, spouse, father, but others are not very informative, such as unknown. But once we identify these matches, we can also infer additional relationships. For example, if two people list the same person as mother, then we can infer that those are siblings. Similarly, if someone lists an emergency contact their mother, and their mother has a sister as emergency contact, then this is that person's nephew. And we could go even further. For example, if someone adds his mother as emergency contact, and the mother has a spouse, we can ask if this is this patient's dad. Overall, including provided and inferred relationships, we identified 3.2 million relationships at Columbia, 1.5 million relationships at Cornell, and 2 million relationships at Mount Sinai. The majority of the relationships are from the nuclear family, such as parent, child, sibling, and spouse. And overall, these relationships represent more than 595,000 distinct families. This is a pedigree, which is the classical representation of family history. In a pedigree, men are represented as squares and women are represented as circles. The patients that we have electronic health record data for have a solid outline around them. The ones that we don't have data for have a dashed outline around them. Some of our families are as big as this one, including up to 4 generations. When looking at the population represented in the families we now have access to, we can see that these families cover a large area around New York City. Here is the colored map that is based on the number of patients with relationships by zip code. The darker colors represent a high number of patients with up to 65,000 patients, and the light yellow represents zip codes where we have fewer patients with relationships. So you can see it covers a very large area around New York City. We evaluated these relationships using clinical data. So both at Columbia and Weill Cornell, we have something called the mother-baby link. It is a link manually created by clinicians for every newborn. It links the baby's chart to the mother's chart. We have about 40,000 of these links in each one of the study sites. And we used these links as gold standard to evaluate mother-child relationships that were inferred from electronic health record data. We achieved about 96 to 98% positive predictive value, both at Columbia and Weill Cornell, demonstrating the quality of the relationships. To evaluate other types of relationships, we used genetic data from Columbia, from IGM, and from Weiser, which is a study that was conducted here before. And those patients that consented for data reuse were identified. We identified 302 patients with genetic data available and relationships inferred from the electronic health record. Overall, parent-child and sibling relationships perform quite well, and we have few patients to evaluate the more distant relationships, such as grandparent and avuncular. Similarly, using data from Mount Sinai, we identified more than 1,200 patients that had relationships inferred from the electronic health record and had genetic data from other research studies with consent for data reuse. Our results here were very similar to the ones we observed at Columbia, demonstrating that these relationships are indeed correct. In a more subjective analysis, we also looked at the age differences between, depending on the family relationship, and we found that the age differences were consistent across sites and were what we would expect on a traditional family structure. Spouses and siblings have around the same age, and great-great-grandparents are older than their great-great-grandchildren. So now that we have all these relationships and family data that was inferred from the electronic health record, we wanted to study disease heritability. Heritability tells us how much variability in a phenotypic trait can be attributed to genetics. But why would we want to use electronic health record data to compute heritability? In this case, the phenotypes are clinical phenotypes. So let's look at different traits and compare the published study size to the number of patients that we have with that trait in the electronic health record. So for example, if we look at height and BMI, published studies had about 20,000 patients, but in the electronic health record, we have about 300,000 patients. When we look at diabetes type 2, the published studies have about 20,000 patients, and we have 140,000 patients. And this is true for a number of conditions. And these numbers I'm showing are only from one study site, but if we include another study site, such as Weill Cornell, then we can see here that we can increase significantly the sample sizes. Okay, so before, how did we traditionally estimate disease heritability? We have twin studies that we compare the phenotypic correlations of monozygotic and dizygotic twins. We have studies that look at other pairs of relatives and compare the phenotypic correlations to what we would expect in genetics. And then we have entire families of individuals that methods that use the entire family and use mixed linear models. And those can handle any relationship structure and different models such as AE and ACE. The electronic health record-derived pedigrees, though, are very different from the ones that we find in a traditional family study. Traditionally, a proband is identified, and then the family members are recruited, and their phenotypes are carefully ascertained. So we determine if somebody has a disease or not. And the ones that we don't know, the missing phenotypes, are clearly marked. In the electronic health record, it's a very different situation. To start, we don't really have a proband, and the probability of phenotyping all the family members is not going to be uniform across all family members. So we have something we call uncontrolled ascertainment bias. This ascertainment bias can vary from patient to patient, from family to family. So for example, patients that live close to the hospital may have very complete records, whereas the ones that live far away and came just for a specialty visit will have less data available to us. So we introduced the concept of observational heritability. The observational heritability is the estimate of heritability that is coming from observational data sources, such as the electronic health record. And here, we control the ascertainment biases by repeated subsampling of families. So for each sampling, we used Solar to estimate heritability of the trait that we adjusted for age and sex, and that's a procedure we called Solar strap. Using simulated data, we introduced bias in the data, and we compared Solar to Solar strap. Here you can see that Solar strap produces accurate estimates in the presence of ascertainment biases that vary from family to family, while Solar was sensitive to the ascertainment biases. However, Solar strap does produce estimates with larger confidence intervals because of the bootstrapping. Solar strap becomes more sensitive to bias as the number of families sampled increase towards the total number of families available. We observe that solar strap overestimates heritability when we sample all or almost all families with the trait of interest. Because of this, and this was a simulated study, we use 15 to 30% of the available families to compute heritability. Overall, we found that heritability estimates were significantly correlated across sites. So here you see the data from Columbia, while Cornell and Mount Sinai, then the correlation between that site and the other two sites. In addition, we also compared our heritability results with the available literature. So for example, a meta-analysis that was published in 2015 reported the heritability of height to be 77%. We observed the same results when running the heritability using the relationships inferred from the electronic health record and using clinical data that was recorded in the electronic health record. We don't use, at any point in time, the information from the literature into our model. We just compared the estimates that we achieved to the literature after it was done. So this is what I mean by the algorithm was frozen. So let's look at some other traits and how they compare to previous studies. So for example, depression, we see that the meta-analysis published the heritability estimate of 34%, and we achieved a heritability estimate of 36%. If we look at celiac disease, again, a study published in 2016 showed 75%, we achieved 77%. Glaucoma is another example in which we do well. A published study from 2004 says that the heritability is 36%, and we achieved 39%. Here, and then we have a few examples in which we don't perform as well. So atopic dermatitis, the published literature is 53%, we achieved 42%. And rhinitis, we see a significant difference here. ADHD, we start to see even more problems in which our heritability was way lower than the published literature, which was 72%. Overall, our heritability estimates are 7 to 20% lower on average. And when we tried to study to understand why that might be. So we injected noise into a simulated data set by randomly shuffling a subset of the patient's diagnosis to simulate misclassification, meaning misdiagnosis or patients that are not coded for the diseases that they might actually have. And we injected noise, and what we found is that if we inject 5% of noise, we see that the estimate changes by 13%. 10% of noise reduces the estimate by 30%. So if we think that misclassification is one explanation of the lower than expected estimates compared to a carefully ascertained study. This work can also be used because we have larger sample sizes, we can clear up some controversies in the literature. So 2 previous studies have shown conflicting evidence for the relative heritability of HDL and LDL. The larger of these 2 studies found no difference in heritability when adjusting for age and sex, while the other one found a slightly higher heritability for HDL, but was underpowered to detect significance. We looked at the heritability estimates here at Weill Cornell and Columbia, and we saw that the HDL seems to be more heritable than the LDL. And this finding holds when we account for the use of statins as treatment for the hypercholesterolemia. Overall, we showed that electronic health records can identify novel traits for future genetic study. Here is a list of heritability estimates for multiple diseases in different groups. We can see we have a huge list. Observational heritability can also shed light on diseases where the disease prevalence has changed due to clinical practice. and we have a few examples of those as well. Overall, we computed heritability estimates for 500 traits. Those include quantitative and dichotomous traits from 12 different physiological systems. And we showed that the relationships that are derived from the electronic health record are real. We can use them. We achieved a very high positive predictive value among first-degree relatives, and we were able to validate these relationships against genetic data. It is a valid method for estimating heritability estimates captured by electronic health record without having to do genetic testing. And overall, we found 500 heritability estimates for 500 traits in our study. And with that, I would like to thank my co-authors and collaborators, and I'm happy to answer any questions. Thank you.
Any questions from the audience? Okay, while other people are thinking, I do have a question. I'll see what's your insight. So when you collect electronic health record, they mention about family relationship. I can imagine that implies one layer is certainly the genetic heritability and all that. But the other layer is many of those close related family members probably live together, right? They have shared environmental exposure and what have you. So how do you distinguish which is kind of heritability genetic component and what is coming from the environment? And maybe there could also be, for most of the complex disease, the environment and genetic interaction. So how do you differentiate and quantify?
Sure. So for us to measure disease heritability, we used a model that is implemented by Solar, which is one recently used and actually very well known and used in the heritability world. And it's a mixed linear model, and we used two models. The AE model, which is the one that accounts very little for environment, mostly for genetics. And the AC model has an additional factor to estimate the environmental component. And for each trait, we estimated both. And when we published both of the results for both models. So we tried to account for that. And the way we did it was looking at patients' address and assessing using the mother's address as the household, so that we could account for people that were in the same household. I see that we got.
Yeah,
oh, thank you. Yeah, we got a question now. In one slide, you showed that for African Americans, the rate was 77%, but the rate was much larger than the other ethnic groups. Can you account for the larger range? Let me see if I can go back to that slide. So in this slide, that's when we looked at the heritability of height, and they were all around 77%, which is the.
Can you reshare? I think we lost your slide when you went back. You probably need to reshare the screen.
Oh, I'm sorry. Let me see if I can try to fix that. Okay, can you see it now?
Not yet. There is a little green button at the bottom, share screen.
Okay, let me try to unshare and share again and see if it goes through. Okay, can you see it now?
Yeah. Yes.
Okay, so let me go back to where I was. here, I believe the question is about this slide, in which we estimated disease heritability for stratifying by ethnic group. And what we saw is that the heritability estimate for all groups was very close to the heritability estimate that had been studied previously with using the meta-analysis data. And we see that they're all around 77%. I think the question was in terms of the number of individuals for each group. So every time we do a heritability estimate, as I mentioned, we sample a subset of the families, 15 to 30% of the families that we have, to compute the heritability estimate for the bootstrapping, and then we do that several times, and we take the average of the bootstraps, and then we get the estimate that way. And so when we look at sample sizes, diseases with different sample sizes, they will be basically sampled based on a percentage of what we have available in terms of the number of families. I'm not sure if that answers the question. Let me know if it doesn't.
There are 3 questions from our side. We've sent it to the chat.
Okay. I can't really see the chat now, unfortunately. So if you...
I can read it for you. No worries. So I'll just using the chronicle order. The first question is from Xia Yang. She asked, did you account for factors in adopted children? That's question number 1. Question number 2, I saw on the relationships graph that you showed both great-grandparents and great-grandchild. What are the differences between the two? And the third one is for height. Was there a range that constitutes a heritable height? So that's the 3 questions.
Okay, so the first one, it is a limitation of the method that if we have somebody that is marked as child in the emergency contact and they're an adoptive child or an adoptive parent, it is a limitation of the method. We can't differentiate unless it's specifically written that this is an adoptive parent or child. So it is one of our limitations because we're using existing clinical data, and emergency contact is a social construct, not really a genetic construct. Patients often will refer to their adoptive parent as parent and vice versa. So those we can't really differentiate and is one of our limitations. The second question that you mentioned, could you refresh my memory, please?
Sure, of course. It's asked, they saw relationship graphs that show both great-grandparents and great-grandchild. What are the differences between those 2 groups?
Okay, so they're just the opposite relationship. So if we look in a, let's see, I'm trying to get to my family structure to show you. Hopefully you can still see my screen.
Yes,
and
we can.
When we look at these relationships, we really have to compute for both the grandchild, grandparent in a traditional pedigree would both be represented. The relationships are simply the opposite. So every time we have, for instance, parents, the opposite will be child. When we have grandparents, the opposite relationship would be grandchildren. And that's necessary for us to build our family trees and make sure that those trees are complete. So that's how we assess. And on the age differences there, you can see that are basically the opposite relationships. So if we look here, you will see that great-grandparents are the opposite of the great-grandchildren relationship.
And the third question I'll repeat is for height. Was there a range that constitutes a heritable height?
Well, height, so the higher the heritability estimate, the more the heritability gives you a sense of how much we can explain height based on heightened in this example by genetics. So what we can see is that what we know is that the genetic component of height, it explains 77% of that phenotype. And we observed exactly the same. So the higher the heritability estimate, the more you're explaining a trait based on genetics.
And since you're on this slide, it's wonderful because Monica, who asked the initial question about the wider range for Black and African American, actually is talking about this range being wider. So it's
okay. Yeah, so she's talking about the confidence interval, I'm assuming, and why it is wider. It is because it is a population that we have fewer patients in our population from the research study. And so when we sample them, because we have fewer families, we capture in the confidence interval more of the extremes. And we observe that for traits that are like very rare as well, which is why the confidence interval is wider than the other populations. Our population has a very high rate of Hispanics, for example. So that's why we see that the Hispanic confidence interval is very similar to the overall group.
Okay, and I also have a question from Bing Chen's lab. So I'll talk about, there are a couple of questions. So the first one, since all three hospitals are in New York, would it cause bias or be used to explain the inconsistency with the literature?
I think that the inconsistency with the literature for some traits has to do more with electronic health record documentation. So for example, in the study, we didn't use data from psychiatric hospitals and nor from psych visits and psychology or psychiatry. And we know that, for example, mental health diseases are poorly documented in the electronic health record overall. So if I'm moving back to this example, for instance, ADHD, we know that in the electronic health record data, this wouldn't be documented very well traditionally. There has been several research studies looking at the data quality of electronic health record data. And we know, for instance, that mental health diseases are not very well captured. So I think that what we see here, it's more about the challenges with the electronic health record data rather than because all the hospitals are in New York. My expectation would be that we would observe something very similar if we were to use a similar data set from hospitals elsewhere.
Wonderful. And the next question comes from Bing Chen's lab is asking whether the EHR system implemented in all three sites are from the same vendor or different.
They are from different, well, so Columbia uses Allscripts, and Cornell also uses Allscripts. Mount Sinai uses Epic. So this method can be applied in different, using data from different electronic health records, and we were able to do that in the replication at Mount Sinai. And the versions between Columbia and Cornell are also different. So even though they're the same vendor, they're different versions of electronic health records. And we were able to run the study in all three sites.
Great. And there is a question from Zhong Wang. He or she asks, how many records are needed for reliably inferring heritability? Will more samples help reduce the confidence interval?
When we have higher sample sizes, it for sure helps us with the heritability estimate. It's hard for me to give you a number in terms of what's the minimum needed, but overall we observed that the traits that we have more data is we can achieve a better estimate. It has to do not only with the amount of patients with that disease, but also how well that disease is documented, the amount of missingness that we have on those families, in terms of the clinical data. So it's hard to give a number, a specific number, but yes, overall we saw that the traits in which we had higher sample sizes, we were able to compute disease heritability. So some of the other traits that were really rare, we were not able to achieve an estimate and converge on an estimate that we could use.
And the related question from Jia Yang is, would it be possible to use all data, not just subsampling?
So the reason why we don't use all data, and I'll go back to that slide here, this one. So this particular figure was built based on a simulated data set, and we were simulating a heritability estimate of 50%. And what you see is that if we sample all families that have all families that we're looking at, then basically what we're doing is computing solar, but instead of once, we're computing the exact same thing, say 200 times. And we saw before that if we don't do the bootstrapping, because the data is not clean, just like a traditional genetic study would have patients clearly marked and they would clearly know which ones are missing. We don't have that in the electronic health record. So what we observed is that when we do the bootstrapping, so when we use Solar Strap, the estimates were robust to noise and that we had in the data, whereas the Solar, which is a traditional estimate without bootstrapping, were very sensitive to these biases. So that's why we bootstrap, and then we select a subset of the families, because we observed that if we select all or almost all, then our estimates are not as close to what they should be. So that kind of accounts for the variation, and we believe that this can be explained by misdiagnosis and issues with the data in terms of biases and missingness and so on.
Thank you. Wonderful. Any other questions from the audience? You can either type in the chat box or you can just shout out. Unmute yourself. Going once, going twice. Okay, so I guess that's it. Let's thank the speaker again. Thank you so much for your support for DASHU and giving us this fascinating opportunity to learn about EHR and big data's application in real clinical settings.
Thank you. Thank you very much for having me.
Yeah. Okay, so we'll touch base. We'll all reconnect in our next event. Stay tuned.
Okay.
Thank you, everyone.
Thank you very much.